Okay, now recording. All right, so ooh, I'm not gonna have my iPad do that. Okay, so we're gonna be spending our first chunk of time this morning, and I imagine this is probably gonna take us up until probably COVID testing time, maybe a little bit before, so we'll see where we get to. We may start in on particle imaging, or we might wait till after lunch. But to start off today, we're going to talk about the link between particle properties and things like size, composition, shape, internal structure, packaging, how those constituents are put together within the particles, and their inherent optical properties. So mean theory, which you've heard about now in a couple of different places and lectures so far, gives us a prediction about particle IOPs. Going from their size and their bulk composition, which we represent as their real and imaginary refractive indices, their complex refractive indices. And it's valid for homogeneous spheres with collimated incident irradiance on them, right? So it's valid for this very idealized situation that may or may not actually correspond to what we see in the ocean. And we're often interested, yeah. A homogeneous spheres would be spherical particles that are throughout the sphere entirely composed of the same thing, the same optical properties throughout the whole sphere. So there's not like an outside that's different than the inside, right? Or say organelles inside a cell, right? Of different composition, different optical properties than the rest of the cell, right? So that would be a heterogeneous composition. Well, we can talk. We'll talk through a little bit of the types of particles that we actually encounter in the ocean, but homogeneous spheres are sort of the simplest possible particle to move along, right? The sphere is a simple shape, and we're just saying throughout its entire composition is identical. So we're often interested in an inverse problem, right? We see radiometric distributions, we see inherent optical properties. We want to know something about particles that generated those observations or those optical characteristics. And mean theory is one way that we can get at that in a forward direction, right? We can start with particles that we model in a certain way with certain sizes, certain compositions, certain refractive indices, and then see what comes out in terms of optical properties. But these model particles are pretty simple compared to what we see in the ocean. So a theme going through this lecture is going to be how well do those model predictions hold up? We compare real observations to mean modeling where there are similarities to be seen. So this is just a schematic that's illustrating mean scattering. And I think you may have seen this exact schematic before, something very similar to it, so I won't spend a lot of time on it. But mean theory is basically assuming that the incident wave is collimated, right? It's all going in exactly the same direction, it's all in phase, and it's landing on homogeneous spherical particles. The cartoon here is not even a spherical particle, so we're already kind of departing from our from our assumptions of our mean theory. So particles that actually scatter and absorb in the ocean might include things like phytoplankton. Are any of them spherical?
The cockroach is kind of spherical.
If you like back out sort of a, yeah, we'll dig into the details of particles here in a little bit. But yeah, they're not exactly spheres, are they? And they're not homogeneous either, right? They have lots of small substructures, they have different shapes, they have corners, they can be oriented differently in the water. Okay, so they're actually much more variable. We've also got non-algal particles, both organic and inorganic ones. So the upper left-hand figures are just showing some electron micrographs of inorganic particles, sand, silt, and clay. So we're just looking at, actually I take that back, the sand one is optical, and the other are electron. But you can see there's some pretty irregular shapes associated with those. We've got non-algal particles, some of which can be quite large, aggregates made up of many smaller particles, also not spherical, not homogeneous. Okay, so what are some particle properties of interest that we might actually want to know something about? Think about your scientific interest. Okay, suspended sediment. So that's a type of particle that we might be interested in. Size. Okay, so just particle size. What are some things that stem from size that we might actually, might be driving us to ask about that? Density.
Sinking rate.
Yeah, sinking rate. So those two are important to people who are curious about sinking speeds. Particles in water. Shape. What do shapes of particles relate to?
The
Structure, like ecosystem structure. Okay, so shape might be some might be a way of getting at, for instance, the identification, right? What taxon is it? And then that might bring us to ecosystem structure for identifying things. Carbon, yeah, that's a big one. So what is the carbon content particle? Yeah. Composition. When you say composition, what are you thinking about specifically? Okay, so the inorganic composition too. So inorganic carbon, like calcium carbonate or silica, right? So there's sort of these things that go together with composition. Okay, these are properties. Yeah. Pigmentation. Okay, I'm going to write this over here by ecosystem structure. Pigmentation. Did I spell it right? Yes. Okay. Yep. Refractive index. Okay. So refractive index is one of those things that we might care about it for its own sake. We might also care about it because it's a way of parameterizing all of these other things, especially things like composition, density, right? Okay, so there's a lot of different properties that in their own right we're interested in because they help us get to things, the questions that we're asking about the environment, about the ocean. So we can kind of lump some of these together into different categories that we can interrogate with field theory and with modeling and looking at particle properties through optical tools. So things like size, what's the size of a non-spherical object? We talked about that last week, right? Think carefully, what do we mean when we talk about size of something that is not spherical? Composition. We characterize this optically with the bulk index of refraction, but of course we want to relate that sometimes to things that are much more specific in terms of chemical composition or water contact or something like that. How much does the shape depart from the sphere? Does it have characteristics that we can use to identify, for instance, species or something like that? The internal structure of particles also are going to be really important when we start to think about optical properties, right? Modifications inside the particle. And then the porosity or packaging, I kind of mentioned this up when I was talking about composition too. Like what's the fluid fraction? How solid is it? Is there interstitial water inside? Right? That's going to affect the density of the refractive index. Okay, so the me solution predicting the optical properties of particles with given size and refractive indices. It's just a theory. Just a theory. Sorry. It's telling us something about an approximation of the particles we're actually interested in. So why care? Why is it so useful? Well, first off, it's very useful because it helps us calibrate our sensors, right? So all of our sensors that list backscattering sensors, flow cytometers, right? We calibrate all of these, especially in terms of size, scattering, right? Through use sometimes of physical spherical particles with no refractor reduction size, right? So this theory actually helps us calibrate our sensors and allows us to compare them. We also expect a given signal for different particles of a given size and wavelength ratio, right? So we can make a prediction for what we would expect to see for these model particles. So there's plenty of optics class alumni actually who use Mie theory, even though it is a simplification in terms of the type of particles we see in the ocean, to provide some really important results. For instance, analyzing flow cytometer data and providing a calibration for the forward and side scattering signals off of these, looking at diel cycles and optical properties in the ocean, and then coming up with a remote sensing conversion to get at particle size distribution in the ocean. So these are all alumni from the last 20 years. Okay, so one of the first applications of Mie theory that really reproduced, sorry, I'm gonna get some water, that reproduced these optical constants associated with real particles was a paper by Bricot and Morel in 1986 where they reconstructed the optical coefficients using Mie theory of phytoplankton cultures. So they started off with several the phytoplankton culture, and I guess you can kind of barely see the label up here. This is the imaginary part of the refractive index that's plotted in the spectrum here. So that's a little in prime. I don't know if you can see that very well, which relates to the absorption spectrum of phytoplankton. And this was one of the first studies to break this down into a sum of Gaussians. So you can see that that's what's done here. And they've taken this imaginary refractive index and then used the anomalous dispersion model to calculate how that impacts the real refractive index, which is, I don't know if you can see this, that's a little n with no prime, so that's our real refractive index up there. And then use these refractive indices and a known size distribution for particles in their culture through Mie theory to predict the optical efficiency factors, so those Q factors, right? The optical cross-section divided by the geometric cross-section of a particle gives us Q value. So here what they are showing is that they've been able to reproduce with Mie theory the attenuation, scattering, and absorption efficiencies pretty well for these phytoplankton species, but not necessarily the backscattering efficiency factor here. So backscattering didn't quite work in this study, which we'll come back to a little bit more in a second. Right, but this is an example of taking refractive index measurements and size distributions of a particle and actually running that through Mie theory and predicting optical factors. So we can take these Mie predictions and normalize them by size and by wavelength. And as I think we've seen this before in a couple of places, but that's going to collapse a lot of these mass normalized optical properties down into a simple relationship. So a reminder size parameter here is just pi d over lambda, so this relationship between the diameter of a particle and the wavelength of light in the medium impinging on it. And then You can also compute rho, the phase shift parameter, which you can think of as basically the relationship between the wavelength of light, or the wave of light, the ray of light, I guess, that's passing through the particle, and one that's kind of going around it, right? There's going to be a phase shift there as things slow down going through the particle. So if you plot versus rho, you end up collapsing this mass-dependent attenuation coefficient. And of course, it varies as a function of the real index of refraction. So we've already talked a little bit about how these Meade predictions and models of optical properties and optical properties themselves have this strong dependence on particle size. The summaries here are just showing on the lower plot, we're looking at a particle size spectrum and different categories of particles, and then where they fall in the different approximations, optical approximations that we use to predict their optical properties. And I think we're going to get into this a lot more this afternoon, so I'm not going to spend a whole lot of time talking about these approximations, but there's different size and refractive index and phase shift parameter limits on ways that you can approximate Meade scattering. Big questions, though, are how do size affect optical properties, and how can we use these optical properties to, in an inverse way, learn about size. So another study that used, that compared, I guess I should say, laboratory measurements with computations that came out of Meade theory was a paper by Amanda Whitmire in 2010. And she took several phytoplankton cultures and in the laboratory measured the backscattering and scattering cross sections, also absorption cross sections of these particles, and then compared them with predictions that came from Meade theory. And we're looking at this on the left-hand side of the slide. The upper plot is showing the scattering cross sections, so square meters per cell of different cultures, and those are the red lines. This is just a handful of wavelengths corresponding to the hydrostat 6, although it's continuous lines there. And then the black lines are from Meade theory. So you can see there's a pretty good agreement between the laboratory measurements and Meade theory predictions. The lower panel is the backscattering cross section. And you can see that the laboratory measurements show that there's this enhanced backscattering over what Meade theory would predict. And this is actually the right-hand panel is just a little example that's illustrating that you do see fluorescence contamination sometimes in backscattering measurements. So we're seeing the fluorescence at 680 that's coming out from those phytoplankton cultures. But this is an example of a lot of times we find these situations where Meade theory, given particles of a particular size and refractive index, can reproduce scattering, attenuation, and absorption cross sections quite well. But then that same set of refractive indices and optical properties will underpredict the amount of backscattering that we see. And this was referred to as missing backscattering enigma, and some hypotheses as to why this occurs. There's one camp that, or sort of group of people that think that there could be submicron particles, right, really small colloidal size scatterers in the ocean that are contributing backscattering that we're not accounting for in our Meade models. And then there's also some that think that there's small scale structure within phytoplankton that might also be generating some of this backscattering as well. The sigma is the scattering cross section of a single particle. Yeah.
Which of these two theories do you think has more weight to it, or a combination of both?
I'm going to say there's a combination of both, because I am not a deep expert in this particular field. I think there are other people in the room who will say other things. Yes?
So, I think Captain thinks it's the shape and film structure of phytoplankton. the history goes a little even before that. I mean, significantly before this 2004 papers, when Morel tried to compare remote sensing estimated to BB based on leaf theory by just summing up all the contributions from all particles we knew about in the evolution, he couldn't account for it. There was not enough BB from those particles based on leaf theory to account. And the issue with the small cell microparticle is nobody has measured it. So there's a lot of paper, including recent paper by Jaidong Zhang, showing that that he can invert them from BSF with a variety of different models, including non-spherical submicroparticles. But nobody has measured those particles. It's really hard to measure those particles. It's really, really hard to measure small, tiny particles. But
Barney Balch measured viruses and looked at backscattering the viruses as a source because billions of viruses per known.
and then we're not enough.
And there were not enough viruses, and their backscattering efficiency was so small that you could not account for any of these backscattering.
And Darius has a paper, a theoretical paper, where he uses Cole's number that people have imported, and again, could not account for that history. With those Cole's from Mark Wells.
So there you have it.
And then there's some people who say that the missing backscatter is really just you measure the remote sensing reflectance incorrectly, and there is no missing backscatter. You just think that there is.
But there's data that are suggesting
That there's some.
that there is. Yeah. Okay. So another result that comes out of mean theory is that the backscattering ratio should have the sensitivity to particle composition, the refractive index, and particle size. And we can actually compute the backscattering ratio. So tilde BBP, there should be an equal sign right there that is missing, equals BBP over BP. And you can contour this, or contour rather the refractive indices that generate constant backscatter ratios as a function of the particle size distribution slope. So this is the differential Young's slope again. So we're thinking about this power law size distribution. So this is the exponent on that along the x-axis there. And so this predicts that for particles with refractive indices in kind of realistic ranges for particles in water, right? So 1.02 would sort of be organic, watery, phytoplankton-like particles, and a refractive index of 1.2 would be more mineral-like particles, right? So through this range, we should see backscattering ratios varying as a function of particle size distribution slope as shown here. So this was modeling work from Mike Wardowski back in 2001. In a very comprehensive study that Emmanuel and his colleagues did back in 2004 in an environment that contained a wide range of particle types, so all the way from resuspended sediments, mineral sediments off the bottom, all the way through phytoplankton-like particles, and using a large number of instruments. I want to say 4 or 5 different instruments, right? So found really broad, reasonable results from many instruments illustrating this variability in the particle backscattering ratio that was consistent with these mean-based predictions. So this is a case where we have a lot of dynamic range, right, through all these different particle types, and we see broad, consistent agreement with the predictions from mean theory. Okay, so size, composition. What about the non-sphericity of particles in the ocean? So we know that particles aren't spheres. This is a summary, so the phytoplankton shapes, I think you've already seen that. The rest of the figures on here are a summary from a review paper by Wilhelmina Cabano in 2007, where she took a data set from San Francisco Bay. This was published in a different paper, and is showing that most of the particles that were present in this particular large sample set they went and they measured the aspect ratio, basically the non-sphericity of the particles, and found that most of the particles that were present in that system had aspect ratios. This is the ratio of the long axis to the short axis if you model it with a spheroid, right? That most of these particles weren't spheres, and that the sediment particles in San Francisco Bay that had these high degrees of non-sphericity also had volume scattering functions that were less peaked in the near forward direction than spheres of the same size. So there is this impact of shape on IOPs. We can also see this if we look at list measured volume scattering functions and inverted size distributions for serratium. I looked it up, by the way. I think the last time I showed you a figure like this, a picture of a serratium, I was like, what are those little things? Are they horns? They actually are horns. Serratium comes from the Greek Horatium, I think, which means little horn. So anyway, I learned something.
Awesome.
Yeah. So now, I don't know, I was like, there has to be a technical term for those. In fact, little horns. But the different portions of the cell of this complex shaped dinoflagellate actually generates a response in the list particle size distribution corresponding to these different parts of the shape interacting and diffracting light. And contrast this in the upper panel. So the solid black line is the list size distribution. Contrast this with the equivalent spherical diameter based on the volume and the equivalent spherical diameter based on the surface area of that cell. Right? So there's a big difference. So the shapes of particles do interact with the optical properties in ways that are really different than for spheroidal.
How did they, sorry,
Yeah.
how did they determine what portion of the cell what part of the shape is contributing to each of these?
I think they're just showing that there's a consistency, right? So
Okay.
if you size, you take a microscope picture and you size this thing and this thing and this thing, you find a piece in the list scatter function. Yeah.
Just on the origin of the word, it also, like Triceratops, right? The
Oh.
dinosaur, the three horns.
Yeah.
Yeah, so Sarah was one.
Sarah.
Is that like protein next time?
Yeah, I bet so.
What was the question?
Keratin makes horns. It's a protein that makes.
Oh.
Yeah.
Nice.
Like our little hands on this?
Yeah.
Have you guys like uniforms like your office class? Sure. Anyway, that's a good thought. So in addition to mean theory, right, so we're talking about ways that particle shapes depart from the sphere, and that this actually influences some of the optical properties we measure, like the DSF. There are other models that we can use to model the optical properties for non-spherical particles. They're all computationally quite heavy, even compared to mean theory. So T-matrix and discrete dipole approximations are the two that are often used, and they're limited to particular aspect ratios for particles. But there are methods that are used to do this, yes.
This says T-matrix twice there. Is that correct? Or should one be?
No, that's both T-matrix, yeah.
Both
So it's
T-matrix.
talking about, axis ratio, axis ratios that can be used for different implementations of T-matrix theory. So we can take, there's a lot going on here on the slides, so I'll orient you in a second, but basically, you can model optical efficiencies with these different approximations for different spheroids, right? So squashed spheres or long spheres, oblated spheroids with different aspect ratios. And look at how much this induces departures in their IOPs from those computed for spheres, as was done with the Lana review. So here, there's the two columns of tiles are basically for phytoplankton-like and mineral-like particles. And in this case, gamma on all of the y-axes is the ratio of the optical property for a spheroid versus the ratio divided by that for a sphere. So it's not gamma the slope of the attenuation spectrum, sorry. So gamma used in a different way. So we've got the ratio for beam attenuation, for absorption, scattering, and backscattering. And then the different line dashed thicknesses are just for different aspect ratios of spheroids. And so the take home here is that we see these large departures from spherical optical properties as we vary the aspect ratio of a non-spherical particle in these modeled schemes. And in general, non-spherical particles have larger cross sections compared to spheres. Many.
Yeah, one thing to think about is there's two things. One is as you go bigger, your towards your copy, the optical properties depend on your cross section. And you can see that the differences get bigger as the particle gets smaller. The second thing to take away is that a sphere is an extreme object. A sphere has the least surface area of all convection. So it's not an average shape, it's an extreme shape. It's true for also, I mean, it's true for nutrient acquisition. There's a whole bunch of things that depend on surface area. Where a spherical model is not your average.
Right.
And and as you saw before, in nature, we tend to see non-spheroidal as the norm. So it's good to remember, even though we will use it in theory, it's useful, etc. Just remember that.
Yeah.
The assumption is extreme.
That's a really good point, and one I didn't make explicitly at the beginning. We were like, we model spheres, but nothing is a sphere. But that is exactly right. Like there's spheres is super extreme in its properties. So if we have non-spherical particles, then it means that they can have different orientations in space, right? It doesn't matter for a sphere, but for spheroids or anything even more complicated, they're going to have an orientation. So does orientation matter? Well, here's some polar scattering diagrams, some volume scattering functions for different spheroids oriented in different directions with phytoplankton, small phytoplankton-like optical properties, and wavelength of 0.5 microns. So you can see there is a difference, right? There are differences in how the incident light interacts with the particle, depending on its orientation in space. And now with holographic imaging technology, we can actually start to get at this a little bit in situ. So this is data from a 2020 paper using HoloCamp to generate in situ holographic images of diatom colonies. So the upper left panel is just an example holograph where you can see these chains all oriented horizontally in space. And what they did was they profiled this sensor. And if you just ignore for a second the colored lines that are superimposed on the color contour figure, what you're looking at is a profile. And on the horizontal axis is the angle that the diatom chains are oriented relative to the horizontal, measured from this holographic profiler. And then the concentration of those colonies is given in the color. So basically what you're seeing is that there's this high concentration of diatom. Ooh, that's red, is a bad color to use there. It matches the line on the plot. Hold on. It'll change. Okay, so here we're seeing this thin layer of diatoms that are horizontally oriented and it's sort of high concentration. And so what they did then is that they modeled how the absorption coefficient would change using a geometric optics model of light bouncing around inside a diatom cell versus as a function of orientation. And depending on the chlorophyll concentration, so this is the black, orange, and red lines now superimposed on the plot paper, modeling how much might AC get affected by the orientation of these cells. So this was sort of the hypothesis that they put out in this paper, is that maybe these cells would preferentially be orienting themselves at certain places in the water column in order to get more light. But anyway, so we can see that not only does orientation matter, but it might actually be going on. Preferential orientation happens right in the environment. Yeah.
So does that mean that when you calculate chlorophyll from absorption, we need to take into account shape orientation?
If you're calculating it in a way that preserves the orientation of the cells in the environment,
Okay.
right? So if you're sucking it through an AC9 or an ACS, probably not. If putting it on a filter, probably not. But if you're inverting absorption from water leaving radiance, maybe? That would be the question I would ask. I don't know if anybody has gone there at all, right? Yeah.
So one gives you an example from nature where you see it. It's always, they always go to the same place you see it. It's east, south, north, south. So it's amazing, it's beautiful, it's a tightly, very stratified environment. How generalized the whole species, we don't know.
They did find it was consistent with turbulence modeling, right, for the type of shear that they were seeing in that water column. So maybe you could physically find.
All I'm saying is that it's a special environment that they've not seen in other places yet. So it's wonderful, it's interesting, it's stimulating, but that doesn't mean that everywhere else we have to worry about. We might need to,
But if you think about how particles flow through these meters, right, which is in a shear flow or laminar flow, are they randomly oriented or are they lined up? Are large linear featured phytoplankton, how are they going to line up in the flow, right? And the ICV or flow cytometry, we tend to align with the laminar flow. The question is how laminar is an ACS and are they lining up? And are you measuring the absorption through the long axis or is it randomly oriented to short axis? But 0.08 is, the absorption out there is huge, so 0.08, they didn't do it as like a percent total.
Right, small.
Yeah, but if it's increasing by 25%.
Yeah.
What do they do in nature? Yeah.
And the tools to get at this are really new, right? Like only in the last 10 years have they even been commercial, and they're not in widespread use. So, so the kinds of observations that might lead to, patterns here, we haven't got a lot of those. Yeah.
There was a time where 3 layers were very much in growth.
Always in this place.
Because they're very special, they're amazing, they're very predatory, the whole variety of things in those layers, ecologically, they're always very, very interesting. But it's really hard to find them in many
So thinking now about how the internal structures within a cell might influence its optical properties, we can look at an early study that was modeling the relative VSF, right? So this is related to the scattering phase function of 3 different modeled structures that were meant to sort of simulate scattering from different parts of a phytoplankton cell, right? So this is going to, I'm thinking about whether I should try and outline this on the plot to make it easier to see, and I don't think I'm going to. But starting from the bottom, the dashed line here, all right, dashed line, this guy here, is meant to simulate a scattering from a hollow sphere, which would be standing in for the cell membrane, right? The outer shell of the cell, if you will. And then the dash dot line, the middle one in the legend. Here, it's down here, simulating the scattering off of the interior, right, the body of the cell. And then finally, in this paper, they used a coded sphere model, which would be the interior of the cell plus the shell, the sphere around it, and the scattering off of that entire body. So this would be this one, right, the one that bounces around. And so what we see is that the cell body is dominating the scattering in the near forward direction. So if you follow that blue line up, you see that's the one that's the most peaked in the near forward direction. And the outer shell, so the cell membrane, if you follow that up, that's much less scattering in the near forward direction, but dominates scattering in the backward direction. And then the coded sphere model of the volume scattering function of the whole cell, right, incorporates both. So this was one of the early results that suggested that by modeling the structure, right, the membrane and the interior of the cell, you can generate scattering properties that are different than either of the parts by themselves.
Does that depend on the phase somehow? Is that why it's oscillating like that?
Yeah, so we're looking at the scattering phase function.
Or like the phase of the light itself?
Yes.
Constructed and destructed interference.
Yes.
That could happen if you do more specific size. Right. You do a combination and all the lines are.
Yeah.
It
Well-
can be slightly shifted in an angle, and then when you put them all together, there's dips out.
We'll do this afternoon, right? Yeah, so you'll be able to make models of this. Yeah.
In the sphere, do they define the two materials or substances by just defining different refractive indices? And then, so did they all look at a difference between whether the outer one had a higher index, or the inner one had a higher index, or whether that would change?
Can I walk you forward into more recent studies that were the tune this? Okay. You anticipate me. This is a newer paper where they are modeling, in this case, it's the backscattering ratios. We're not looking at a phase function, but we're looking at the relative amount that's in the backward direction, right? So it's from that. But looking at the backscattering ratio for modeled particle populations with different size distributions, and taking a homogeneous sphere with optical properties, or population of homogeneous spheres with optical properties consistent with the oligotrophic ocean, a bloom situation, bloom of a phytoplankton species, and then coastal waters with lots of high index resuspended sediments. And then using a coded sphere model to adjust the model of backscattering fractions from those same particles, those same refractive indices. So what they found was, in particular for phytoplankton, right? So for the oligotrophic ocean case and the coastal bloom case, using this coded sphere model increased the relative amount of backscattering significantly. Yeah.
What is their x-axis?
So the x-axis here is eta, the slope of the particle size, the power law particle size distribution slope. Yep. So many more smaller particles and many more larger particles would sort of be your qualitative axis label there. But for eta equals 4, which would be kind of our Younga type power law size distribution, right? Here they see from their reference cases these large increases going up to the dark red and green dashed lines. Sorry, these are not good color schemes here, but if you go up to the dashed lines above the solid reference case lines, you're seeing the influence of using this coded sphere model. And in even another recent study, in this case, they took measurements in the environment of backscattering and beam attenuation, and then tried to reproduce those measurements with a me model using sort of realistic-ish refractive indices and size distributions. And they were able to do it pretty well with a me model for beam attenuation, right? So in the upper left panel here, right, plain me model is able to reproduce beam attenuation relative to the measurements, but not backscattering, which is the upper right panel. However, if you use a coated sphere model and tune the inside of the cell and the outside of the cell to reproduce the observed beam attenuation, you also reproduce the observed backscattering. So this is another result where modeling the structure of cells, modeling the outside of it and the inside of it separately seems to increase the agreement with observations. All right, these are slides that Emmanuel put into this lecture 2 years ago, and I have to give him credit because I think they're, I don't know, not funny, that's the wrong word. They're telling, they're appropriate.
Frank Boren is a character and first of the book. But it's a pleasure to read his books because he does not shy from providing. There are other people are afraid to do it. And this is what he says here.
Yeah. So, he basically is saying what we have been seeing, right, in the results in the papers that we've looked at so far in this lecture, that me theory doesn't reproduce observed backscattering. And then goes on and says, when criticized for using me theory, where its applicability is dubious, modelers sometimes respond that although they know me theory is inadequate, it's the only game in town. Better to do wrong calculations than none at all. Modelers have to model. Suggest an alternative. It's called not modeling. Not modeling until adequate methods are at hand. So it is, says it right there. Anyway, so this is sort of something to keep in mind as we continue to use me theory, which is very useful. And as Colin's dragon footprint slides have illustrated already, we're trying to do a really hard thing backwards in remote sensing and optical oceanography, right? We're trying to infer something about really complicated, heterogeneous, almost infinitely diverse particles from optical properties. It's hard to go backwards. We need a way to go forwards. And me theory is one of the tried and tested tools to do this. But there are different ways of modeling these particles. And so it's worth trying different techniques and thinking about that. This is another study that is using modeling of optical properties, in this case, geometric optics modeling, and looking at just physical ray tracing, physical optics, on little models of coccolithophore parts, right? Of the coccoliths themselves, modeled here, like a little wagon wheel, or a coccolith disc attached to a fragment of the shell, to try and understand whether the small scale internal structures that you see in those little coccoliths matter in terms of backscattering. So this was one of the early investigations into, where is the missing backscattering in the ocean coming from? Basically, Howard Gordon modeled pieces of coccolithophores either as a disc attached to a fragment of a cell. That's what this upper diagram here is showing you. This is what it looks like in cross section, or just as a homogeneous disc with no structure at all, or using a discrete dipole approximation to model scattering off this wagon wheel-like structure in the lower diagram there. And what you see is that for all of various thicknesses of these cap and disc configurations, there was not really a whole lot of difference from a physical ray tracing model of the scattering cross-section per particle. And in the lower panel, we also see the same is true for just a plain old disk. But this wagon wheel type thing did induce a departure. So that would be this line here. A departure from this physical optics model only when the features, these little tiny bands within the wagon wheel, when those started to exceed about 1/4 of the wavelength in size, then he did start to see some departure. So there was a little bit of nuance, I guess, in this model, but for the most part, even coccolithophore disks seem to scatter without a whole lot of influence by those small scale features. But he basically took this wagon wheel and sort of increased the complexity and the size of those little wedge-shaped features in it more and more and more until you could see when it started to matter. Let's see, where are we with time? Oh, excellent. Okay. So another aspect of particle structure in the environment is aggregation. So when you think about, I guess I showed some photos of aggregates on the first few slides, right? We're talking about agglomerations of different primary particles to form an aggregate. These are hot topics to those of us who like to think about the biological pump and sinking particles and big balls of poop in the ocean, et cetera. But aggregation, if we're thinking about how they form, right? Aggregates are going to form, the rate of aggregation will depend on the concentration squared, because you have to have subparticles, primary particles encountering each other. They can encounter each other at very small scales through Brownian motion, at larger scales through differential settling. So if one particle is falling slow and then another particle is falling fast and runs into it, right? It would be sort of the very simple description of that. Turbulent shear, and also biological processes, which is something that I think about sometimes, but hasn't worked its way into a lot of the aggregation models yet, which I'll show you another slide on that in a second. Aggregates sink faster than their component particles do by themselves, right? Stokes' law, which is also for spheres, right? So Stokes' law varies if you have different drag on non-spherical particles, but that's a different class. But to first order for spheres, the sinking rate of particles, one of the things that we were interested in on our first slide, right, goes as the square of the particle diameter. So aggregates sink much faster than their component particles do. And then if they sink too fast and if shear is high, then they can break up, right? So there's a lot of complex processes that are going to form and take apart aggregates in the marine environment. This is one set of photos from a coastal environment above bottom, and it's in 12 meters deep. And both of these photos were taken within a day. So we're basically looking at how much aggregate size and concentration can vary just through the course of a tidal cycle, showing some of the variability there. So how does aggregation influence IOPs? Emmanuel and Wayne Slade, who was a student at the same time as me a while ago, did some interesting studies, both modeling and experimental, looking at the effects of aggregation on inherent optical properties. So they started off just modeling, right, and thinking about some there's a few models out there that predict how the size and the fluid fraction of aggregates scale together in the marine environment. So basically, the bigger an aggregate grows, the more of its volume is composed of just fluid. And you can think of it as, you know, a large aggregate might be more like a stretched out cotton ball. Like if you take a cotton ball and just start like pulling it apart till it's big and fluffy and almost see-through, compared to a smaller aggregate that might be more densely packed that you can't see through, right? So that behavior, that tendency does something interesting with the optical properties, which is that if you take the beam attenuation of aggregates and normalize to their mass, which is what we're showing on the y-axis of the plot here, we see that as aggregates grow larger and larger and larger, their optical properties normalized to mass actually don't change that much. Certainly not as much as we see with solid particles. And we model optical properties with mean theory off solid particles, right? So there we see orders of magnitude decreases in beam attenuation normalized to mass as a single grain grows bigger and bigger and bigger. But if we have a particle that instead is made up of aggregates with high fluid fractions, as that aggregate grows bigger, beam attenuation doesn't change all that much. And this makes sense because aggregation is conserving area, not volume. So this is a modeling prediction of what optical properties ought to look like. And a lab experiment shows something similar. So here, I'll walk through the panels top to bottom. Basically, we took a sink full of suspended clay particles and then added salt. So if you have a bunch of clay particles and you add salt, you're going to cause them to electrostatically attract. They're going to start to stick together, and you're going to grow aggregates. And so over the course of this experiment, from 0 to 3.5 hours, in the upper panel, you see the blue dots, the suspended particulate matter concentration decreases because the clays aggregate and then they sink, right? And you're observing this sort of at the center, I guess, of the sink, the center depth of the sink, rather. So the concentration decreases as things fall out of solution, and the average diameter of the particles is inverted from the list VSF increases, right? So you grow aggregates, they sink, they fall out.
How much salinity are they adding into your clay? A part per million, 30 parts?
Probably 10 at a minimum, I would guess.
Probably 10 to be, this is completely artificial. We're using some clay, I don't know if it's scale and a bisol, not even NACL, something that causes the
Okay.
So it's an analog, but a poor analog, it's just something that was easy to make and reproducible every time we ran it at the same result.
In a real estuary, you would start to see that effect. You know, you don't need to get the full ocean salinity.
Oh, yeah.
Effect. You can see it much
The
higher.
no full PSU, no full PSU in the estuary.
Is this flocculation? Is that what this is?
This would get flocculation as sort of a subset of aggregation. But yeah, flocculated clays.
But just that.
This is what your town is doing with your sewage. You add alum to sewage to get the stuff to come and fall into the water. This
This is how I collected river sediments from my PhD. Like a garbage can full of dump a bunch of salt.
This is also how they propose to clean up halves, toxic halves, if you add clay to salt water and it aggregates fully and it rapidly clumps up the harmful algal phytoplankton or the harmful phytoplankton and sinks them out to the bottom.
So anyway, in this experiment, that's what they did. Aggregated clays with salt. So we see an expected evolution of particle size and sinking in the top panel. In the middle panel, we're looking at just the straight up IOPs, so the volume scattering. Volume scattering function is the red diamonds, and the beam attenuation from the list and beam attenuation from the AC9 in green and blue respectively. And all three panels have this average diameter from the list superimposed. So we can see, as we would expect, as stuff falls out of solution, the IOPs decrease, right? But in the bottom panel, now what we're looking at is those same IOPs, but now normalized to the suspended concentration at the depth where the sensors were measuring. And in fact, particularly for the volume scattering and the list beam attenuation, we see pretty constant properties, right? Which is consistent with that modeling study on the last slide. The AC9 also changes a little more, it drops a little more. Why would the AC9 beam attenuation, mass normalizing attenuation, decrease more than the list? In the same water, yeah.
What was that normalized to again?
They're both normalized to the mass concentration of particles in the water. So basically, if you take the second panel colored symbols and divide by the top panel blue symbols, you get the bottom panel colored symbols. Yeah.
Angle of acceptance?
Yeah, so the List acceptance angle is much smaller, so it's gathering more of the near forward scattered light that we would expect to be enhanced as particles grow larger. So the flocculated particles grow larger. So there's an acceptance angle effect. Here. Finally, they did a study where they tested this off the dock in the environment. So here we're looking at two lists, one of them with a pump in front of it breaking up the particles, and the other one not. So we see that the red list and the blue list, so the red list is the one that has the pump in front of it. So breaking all the aggregates up into primary particles, and the blue list, the sample chamber is open to the environment. So we see there's this difference in the mean size at the very end of the experiment in the last couple of hours. Both lists were left open to the environment as a control. So you can see that once that happens, then both lists agree on the mean size distribution. While the pump's there, these primary particles are getting broken up. But in spite of this, in the upper left panel, we see that the beam attenuation is the same or nearly the same. There's a small difference, but they're almost the same, right? Which is reinforcing this idea that aggregates have similar IOPs, basically, as their constituent particles. Because the aggregate is mostly fluid and its surface area basically is all interacting to light in the same way the primary particles would be. You do see a difference in the volume or in the phase function, the scattering phase function, right? So the unpumped open list, the one that has all the aggregates, has this enhanced scattering in forward angles, right? So you still see a difference in terms of the diffraction around the particle. I don't remember what the lower right-hand panel is.
Unpumped.
Unpumped, sorry?
Both of them unpumped.
Both of them unpumped? Oh, okay,
Just to show
yeah.
that they're the same.
Got it. So this one goes there. So we see this modeling in the lab and in the field. And it has important implications for when we're looking at properties of large particles and aggregate particles.
So I've heard that when you are taking phytoplankton, the pump you use can matter for whether you break up the cell or not.
Yes.
If you break up the cell, would you expect to see a similar, you don't have a change in CP, but you have a big effect in B, or would it be different because we're breaking up a cell as opposed to an aggregate?
I would think it would be different because we're talking about breaking up a cell, right? And if I think of a cell or a chain of cells, so I think a lot of when you're thinking about what kind of pump to use, for instance, in your inline system, you don't want to break up aggregates, but you also don't want to break up phytoplankton that are linked up in chains or structures of that sort, which may not scale in the same way as aggregates do.
Yeah, there's a nice paper by Ivona where they actually compared it. So Ivona that paper is Economics Express, where they compared a gentle pooling of water from the rosette to water in the inline system and found over 30% differences in cell counts and in IOPs. So it's something that on any cruise you go, you want to do. So you want to compare measurements that are done with the rosette, or you take a bucket versus the inline system to convince yourself the inline system is not insignificant.
And people think about the diatoms and sort of breaking the spines off and breaking the chains, but you also have to think about the softer side of them. Some of them are just burst. You don't ever see them in inline systems that have rough pumps. And you see them in minuscule bottle samples. And so if you have an ISTV, it's a great tool to look at individual plankton types and compare who's disappearing in a pump system.
And Steve Atkinson had really nice follow-up paper with this method where they actually have a control chamber in the turbulence, and then they measure down that chamber the change in sizes. So they're actually looking at how, depending on the turbulence in the environment, am I changing the size of, am I desaturating? How much energy does it take? So they control the shear. Here we just do a sequence problem on the intake. And we not control anything about the shear, we just want to break. He actually wants to break them in different intensities so he can tell something about their properties. Because people who study aggregation in technology, for them it's really important how steep you fall. And by varying that rate, you can study the steepness.
All right. 2016 for Appleson or for Sesame? Thank you. Ooh. I should have an equal sign too.
And an integral.
So,
And an integral.
and an integral, yes. Let me do it so that we can do both. There we go. I think that's everything that should be in there. Okay, so take home here is that when you're dealing with particles in tissue, you want to take into account aggregation. You can't just assume that the primary or that the primary particles and the aggregated particles are just a sum over the constituent particles. Aggregation is important because of settling velocity. It's important because of aggregated particles and their influence on the underwater light field at depth. And just at the end, I stuck in this slide about biological aggregation processes, which I think biological aggregates, as I call them, receive a lot of attention in optical oceanography, in part because a lot of times we find these well below the depth where the sunlight gets to, so you're not necessarily going to see them in remote sensing. But things like fecal pellets, appendicularian houses, these gelatinous mucus and feeding structures that incorporate lots of primary particles and are thought in some environments to contribute a lot to things like carbon export. Might be sort of an interesting thing to think about. This is a manual scuba diving, all the 2 tons back. Also, a thing I left in because it's funny. Sorry, it's impressive.
It's actually buoyant.
So there's a lot of diversity in particle types that we find in the ocean. Modeling, remodeling can get us a lot of the way there. It certainly helps us do the forward connections that we need to complement the inverse questions that we're asking. But there's still a lot of work to do in terms of accounting for the different shapes, internal structures, aggregation states, etc., that we find in particles. With that, it's 11 o'clock, so we're about an hour in right now. We have COVID testing starting at 11:30. So I would suggest we maybe take a 5-minute light stretch, grab water, etc. Come back, we'll start on particle imaging, and then we'll stop when we have to stop. And finish up.
There should be coffee, fresh coffee.
Get some coffee.
Question about the aggregate sinking.
Hold on one second to stop the recording.